Well, thank you. So I'll just get right into it. And my slide really looks like Carson's introductory slide, rotated about 90 degrees. So why do we do metabolomics? Well, the metabolome is downstream from the genome, the transcriptome, and the proteome. To some degree, encodes that information. It's also a phenotypic. We appreciate that the metabolites are proximal reporters of diseases like type 2 diabetes and chronic kidney disease and inflammation. But the metabolome is also influenced by diet, microbiome, lifestyle, and environment as well. But measuring the metabolome is a significant analytical challenge. The first reason is that we actually don't know the number of metabolites that exist in a biological specimen. The second is that the concentrations of metabolites that we find in a sample may range, or in a biological tissue, range from, say, 1 molecule in a cell to millimolar concentrations, but our best analytical methods only measure a narrow window, maybe 4 to 5 orders of magnitude, linear dynamic range. A third challenge relates to the differences in physical properties among metabolites. So the key one is really the polarity of metabolites. So we'd like to measure everything from polar compounds like fatty acid, or like organic acids like lactate, to lipids. And chemical stability is another issue. Some compounds don't survive our extraction processes, just they're chemically labile. So in short, really, we have to use multiple analytical methods if we're going to attempt to get full coverage of the metabolome. The other point I would add here is that unlike genomics, where you might be looking at kind of qualitative information, the presence or absence of variation in a gene, in the case of metabolomics, the metabolites present in most people, what we're trying to measure are differences in the levels. So we have to have very precise methods. So one strategy for measuring the metabolome is to use liquid chromatography, tandem mass spectrometry, or LCMS. And this is just an example of our platform at the Broad Institute, where we take a single sample, we'll aliquot it, say we'll start with about 150 microliters, make some small aliquots, and then do separate extractions, and use methods to measure things like lipids, another method to measure compounds of intermediate polarity, such as free fatty acids, oxidized lipids, and then a couple of methods to measure polar metabolites. And these data can be acquired in what we call a non-targeted manner, where we measure signals from metabolites that we know the identities of, as well as signals from compounds that we don't know the identities of. We also have targeted methodologies where we focus on specific classes or specific metabolites, and then you kind of dial those in so you only measure short lists of metabolites. And this is what the raw data end up looking like. We have chromatographic peaks where we know the retention time and the mass, and we can integrate the area into the peak. But these data were acquired using a non-targeted methodology. This is the same retention time axis, and these peaks are actually covered by these ellipses here. And in this run, for example, we know the identities of about 250 metabolites, but there are a couple, 2,000 or 3,000 other signals that we do not yet know the identities of, but we can continue to measure reproducibly across multiple samples and through time, and we can track those based on their measured mass and their measured retention times. So we haven't done the UK Biobank, but we have used metabolomics to study incident disease in a number of different cohorts, and quite a bit of work in type 2 diabetes, cardiovascular disease, and cancer. So I just want to show you a quick introduction to what we've done. The first study I worked on when I joined the Broad back in 2009 was a collaboration with Robert Gersten and Tommy Wang. And our goal here was to find early indicators of type 2 diabetes in the Framingham Heart Study cohort, which is a multi-generational cohort. And the second generation participants underwent an oral glucose tolerance test in the early 1990s. There were about 3,000 participants who did the OGTT, and of them, about 200 of them were deemed to be of normal glucose tolerance at the time of the test, but about 200 were later diagnosed over a 12-year period with type 2 diabetes. So the initial study was a nested case control study where we had baseline samples from 189 future type 2 diabetes cases and controls that were matched based on fasting glucose age, sex, BMI, and hypertension status. And at the time, we were using a targeted platform measuring about 250 metabolites. And what we found was that the branched chain amino acids, isoleucine, leucine, and valine, as well as 2 aromatic amino acids, tyrosine and phenylalanine, were elevated in plasma of these incident cases up to 4 to 12 years before diagnosis of type 2 diabetes. And people that were in the highest quartile had anywhere from a 2 to almost, 2 to 3.5-fold higher risk for developing type 2 diabetes. And we could take these data and combine them into a score. So in this case, isoleucine, phenylalanine, tyrosine, people were in the 4th quartile for that score were actually had a 6-fold higher risk for type 2 diabetes. And we validated that in the Malmo diet and cancer study using a similar sized nested case control study. We also examined triglycerides and other lipids, and what we found was an interesting pattern of acyl chain carbon numbers. So if we totaled up all the carbons that were in the fatty acids and the triglycerides and just plotted those cases over controls, we found that people that were incident cases of type 2 diabetes had elevated levels of short acyl chain containing triglycerides that were mostly saturated. And then we could look at other metabolites and find individual predictors. In this case, a compound 2-aminodipic acid, which is a metabolite of lysine, was a strong indicator for risk for type 2 diabetes in both the Framingham cohort as well as the Malmo diet and cancer study. So after we did these studies, we then moved to doing non-targeted methods like the ones I showed a couple of slides ago. And this is just an example of some work we did in the Gen 3 cohorts, so the children of the participants in the last study. Here we had 1,000 samples. We applied one of the 4 methods I had shown. We measured about 5,000 peaks, and we found about 500 of those peaks were associated with key metabolic traits. And shown here are associations between metabolites and HOMA-IR, a measure of insulin resistance. And some of these p-values are quite low. Among these, we also had a number of unknowns that we measured, again, we just know by mass and retention time, that were also associated with HOMA-IR. The Gen 3 participants had also undergone, or at least 500 of these 1,000 had undergone chest CT scans that enabled us to enumerate liver fat, and we found 200 peaks that were associated with liver fat. The strongest, the peak that was most highly associated with liver fat was an unknown, and even after adjusting the regression models for age, sex, smoking, alcohol use, HDL, glucose, diabetes, hypertension, the p-value for association was still 10 to the minus 13. So the question is, what is this metabolite? So normally what we might do for doing a study like this is we'd look up this mass in tables of metabolites, trying to find potential matches, acquire those compounds, and then test them in the method to see if we find matching data. We did that and couldn't find any matches, but fortunately, genomics had been done in the Gen 3 cohort, and it turned out that this unknown metabolite was highly associated with variation in the gene for AGXT2, or in the AGXT2 gene, alanine glyoxylate aminotransferase 2. So the p-value here was 10 to the minus 9. Oftentimes these p-values in such a small study don't reach genome-wide significance, but in this case it did. Fortunately, this enzyme had been previously characterized by others as a multifunctional enzyme that handles a number of different substrates. One of those substrates was asymmetric dimethyl arginine, a compound we do measure, but this product, dimethylguanidinovaleric acid, or it's easier to call it DMGV, was discussed in the literature, but not in the metabolite databases. And it turns out that the mass for this product, DMGV, matched identically to our compound that we found through non-targeted profiling in our study. So then we were able to then make a targeted methodology just to measure DMGV and then do cross-sectional analyses of other people. So looking at people who had biopsy-proven non-alcoholic steatohepatitis, DMGV is elevated in their plasma. And then in a separate group of people who were undergoing Roux-en-Y gastric bypass surgery for weight loss, we see that DMGV levels drop as a function of time following surgery. Further, DMGV is also a strong predictor of type 2 diabetes. The Gen 3 cohort doesn't have many incident cases just yet, it's still relatively young. So we look to the Malmo Diet and Cancer Study again, as well as the Jackson Heart Study. And DMGV, again, has associated with a 2 to 3-fold greater risk for type 2 diabetes. So my goal really today was to tell you that we're able to do projects of scale using metabolomics. So how do we approach that? Well, the first thing to know is that we acquired the metabolomics data in batches. In our lab, it's batches of 1,000 samples. Others may use different batch sizes. But a key thing to doing large studies is to be able to serially collect and concatenate the data from batches as they become available. And so in order to do that, you have to have a number of strategies. One is to have good QC within each batch of samples that you're going to analyze. You have to have strategies for standardizing the data for drift in instrument sensitivity within each batch. So these are mass spectrometers. The sensitivity can drift through time, and a batch, in our case, may take a month to acquire. And then you have to be able to scale the data to be between batches. And then last but not least, probably the biggest challenge for working with the non-targeted data is that you have to have a means to align the unknown metabolites. These metabolites that you don't know the names of, you don't have standards for, you just have measured masses and retention times. And from batch to batch, there's small variations in mass and retention time that complicate aligning these features, because you may have lists of 10,000 unknowns, and you can make mismatches. So the first thing is to do QC and data standardization. There are standard approaches. One is to use internal standards. We introduce those into the samples and monitor those as a function of time. But the key thing is really using some type of pooled reference sample. And we use pairs of pooled reference samples that we insert into the analytical queues at intervals of about 20 study samples. This is fairly common in metabolomics. In our case, we use the first sample to scale the data, remove temporal drift by standardizing the data as a function of time. And then the second sample is used as a QC that enables one to measure the analytical precision of the measurement for every single metabolite and unknown in the sample. The nice thing about if you make enough of this pooled reference sample, you can run it with the entire set of study samples, and therefore scale all of the data across batches. The other thing that's required is to have a workflow that supports the processing, post-processing of the data. So in our lab, we have a cloud-based web server that hosts a number of different applications. One of them runs our data standardization app, and then a second one runs our alignment algorithm that uses some unique algorithms. And I just was going to show an example that indicates the feasibility of doing the analysis. And I have to admit that it represents maybe not quite 2% of the UK Biobank. And the other thing I have to apologize for is that NHS in this case represents Nurses' Health Study, not National Health Service, so I apologize for that. So in this case, I was working with Heather Elias and Walter Woolett, Mayor Stanford, and Fran Grodstein on a project that's ongoing where our goal was to find risk of incident breast cancer and colon cancer in these cohorts. And these were divided into basically 3 projects. Partly it was due to funding, but our goal here was to concatenate all the data together. So we had about 4,000 samples from the Nurses' Health Study, about 2,600 samples from the Nurses' Health Study 2, and about 1,600 samples from the Nurses' Health Study and Health Professionals Follow-Up Study. These were colon cancer samples. And we had to spread these analyses over multiple chromatography columns, each time measuring a couple of hundred, this was with a single method of those 4, measuring a couple hundred metabolites we know the identity of and thousands of unknowns. And once we had the data, we were able to align the data sets, meaning we could align, obviously, the knowns, that's easy, but the unknowns. And so we aligned the data together. And what these are plots of coefficient of variation for each metabolite in the pooled QC samples and the peak area, the raw signal intensity. And these look terrible because it's raw data, so really high CVs. But after you standardize the data, we end up going from having, say, a median CV among our knowns of 24% in terms of the signal intensity down to a 3.8% median CV. And the unknowns, which in this case, or in any non-targeted metabolomics method, include both real metabolites and real biological molecules, but also contaminants and your plastics and solvents and whatnot, where the CVs were much higher, but higher in the raw, 56% median before we standardized the data, and after standardization, about 17%. So we were able to do that across each of these 3 sub-studies and then combine the data. And after combining, again, data from about 8,000 samples, the median CV of 212 knowns was 3.9%. We had about 187 metabolites that had CVs under 10%, and 205 with under 20%. Among the unknowns, the median CV was 12.2%, and we had 1,200 that had CVs of under 10%. And that's kind of the range you really need to be in, because some of the effect sizes that we measure in these studies of prediction are quite small. So just to conclude, working with smaller cohorts, we've been able to identify metabolic profiles that predict incident disease. The effect sizes tend to be small, like 10%, so doing a complete analysis of the UK Biobank would provide terrific power, statistical power. We do have scalable metabolomics workflows in place that are compatible with doing large-scale studies. And the modular nature of our LCMS-based approaches means that you could maybe run one method in one site and another method in a different site, or one method at a time, or depending on one method, if you only have funding that would support one method. And the last thing I'd say is, in considering what to do with the UK Biobank samples to run other just targeted methods alone where you measure only knowns, I would argue that you probably should consider running non-targeted methods that measure both knowns and unknowns to enable serendipity. Thank you.